comes good with this pandemic, lockdown, and all this confusion is the kind of digital invasiveness. We have actually one area where we would have been failing or slow to catch up will be to use the digital resources. Now, digital invasiveness has come into biology also. Even now, as a biologist, I shy from my mobile phones, and I don't really follow the digital, principles in my rule. This is because I always, as a biologist, speak face to face. I talk people with my friends, students, face to face and flesh to flesh, that kind of interaction. But nonetheless, you would find that we have to come of age. We have come of age, and we have a scientist, a holistic scientist, who had actually accomplished himself in a very, very short time into a very powerful researcher, a leading teacher, and an excellent speaker who had been invited to almost all biology departments. Just a little while ago, Sanjay told me that he had brought him to botany department, and somehow I missed the opportunity of listening to him. Now, with digitalization, genomics, molecular technology, diagnostics, bioinformatics, all these things have turned buzzwords. And drug development and personalized medicine. This is something which has come to stay, and this is going to rule. This is going to stay and rule. And we have an expert in Madurai who is well versed on these things, and he is going to talk to us. Before I give him some time, I would like to use a few moments to introduce him. Why? Because he was a student like you. In a very, very short span, he has overtook all seniors like us, and he has reached a very big height. And to motivate you and to recognize what he is, I thought I would catch a glimpse of his personal biography. So he was his biography tells me that he was a student of Paramakalyani Institute. You say that to me, Madurai is a big village, and what is Alvar Kurichi and Paramakalyani, you can think about it. Even though his brother was a famous professor in MS University, he was in Paramakalyani University doing his undergraduate in Paramakalyani College. I think it's Staines College. It's actually, Staines is one big firm, and that's college. And I'd been, roughly around the time he was a student, I'd been invited to give an inaugural address or something like that in one of the science departments. So in 95, he was there. From there, he won the gold medal and came over to Madurai Kamaraj University, where again he joined biology, took the gold medal again. And that was the time that biology was so popular, biotechnology and biology was so good that normally students that easily don't get admitted into the programs. And it's all India kind of admission done for biotechnology and biology. And in that batch, he has actually again excelled and come up with a gold medal. And he continued there, and he was doing his research. Now, the person whom I am introducing is Dr. Professor Ganeshan Kumarasan. He's Kumarasan, that G stands for his dad, I guess. Okay, Mr. Ganeshan Kumarasan, Dr. Ganeshan Kumarasan. So, Professor Kumarasan, okay, he shaped up his research career after he's completed his PhD with stalwarts here, with good professors here. I'm told that he was actually working with Professor Madhavan, who is a well-known fish biology person, and then later PGS. And these two are, PGS is of almost of our batch, and Professor Madhavan is a very soft-spoken person, but two were very accomplished scientists in the microbiology division, and that's where he had actually his research shaping. And from there, he went on to, I think, National Cancer Center, NSU, Singapore. So where he was actually completing his PhD, and then he was actually doing his PDF also. I think he was RA in Agenica Private Limited in Singapore and National Cancer Center. Then for his PDF, he went to US. I think he was with MD Anderson Cancer Center, Houston, Texas. Okay, I had the opportunity to travel through Houston, Texas, and I know Texas is one place which was fascinating to me because my professor, Dr. Madhavan, was a PhD from Texas. Okay, he is now, a few days back he spoke to me. At the age of 75, he is retired now. He was actually, sharing his good concerns. I know the Texas was actually a very good place for biology, and there he did his, cancer research, and he accomplished himself, and he joined the university here back as associate professor, and very quickly he moved on to become the professor. Since 2013, he is the professor and head of that particular institute. And when I said the organizer, Dr. Priyadarshini, that I do not know him personally, she gave me this much bunch of papers. So she gave me homework to work on, and if I have to complete it, I'll take this 1 hour. I don't want to do that. This is because we have a very big scientist there. Here and there, I would like to speak about a few accomplishments. He never publishes in local journals, local in college conferences and all that. His publications are of that level. You should know that. Don't get into the habit of publishing papers in local journals. So he is a model. Okay, he has actually done it. And at least 3 publications I was very impressed. I wanted to share with you. Current trends in biomedical genetics for personalized targeted therapy is the subject on which he is going to dwell today. Now, his publications relating to that is worth mentioning. Targeted therapeutics, which appeared in Cell Research in 2022, is a big thing. And he has actually worked on, he's a medical anthropologist, I would like to call him like that. And he has actually published a paper in Nature. I don't think I will that comfortably hold a journal in Nature in my hand. This is because that was setting the standards. You make one publication in Nature, you've done science. So here is a man so humble, so young. He has published in Nature on a very, very interesting topic, ethics of DNA research on human race. And they were actually giving us a very interesting title, 5 globally acceptable guidelines. So these guys, including this professor, were giving the guidelines for the global research. So published in Nature. Research is his cup of tea. He had made enough and more of publications, about 50 publications in all top journals. He has produced countless number of PhDs. And all of them are doing good, and that's why the biodata is so very thick and tough. And genomics is actually his forte, and therefore he is invited grants from different agencies. We think about grants which will be in a college. We will be talking about a grant which will be 1 lakh, 2 lakhs. Now he is a man who has attracted grants of 3 to 4 crores from one agency. So he is with all kinds of recognition and a very strong laboratory, surrounded by a team of very dedicated researchers, research students. He is an expert in bio-nano. He is good in bioinformatics. He has worked on nanocomposites in bone regeneration, and he was actually one another interesting thing is in biodata he says drug repurposing. He will have to explain this to me personally after. What is this repurposing? I think he is trying to change the paradigm. And he is well versed with the human genome, and in cancer, in at least 3, 4 different types of cancer, he is the man to look for. And I had never seen such a strong biology professor, at least in the past 20 years in Americancollege University. When we were there, entering the services some 35 years back, MKU was actually setting up the trend for the India, and they were all big names. So trained by them, he is truly living up to their standards, and he is the person who is now guiding and leading the Americancollege University. For the next 1 hour or so, he is going to talk to us on perhaps the personalized drug therapy and things like that. Biomedical genetics is going to be the engagement for the next 1 hour. I want you to give a keen, good listening. Feel free. He is one among us. இங்கேருந்து ஒருத்தர் எழுந்திருச்சு என்னுடைய மாணவர் அங்கே உட்கார்ந்துருந்தவர் இங்கே ப்ரொஃபஸராக இருக்கார் சிவரூபன். சஞ்சய் இங்கே வந்திருக்கார். இவர் இங்கே படிச்சிருந்தாருன்னா என்னுடைய மாணவராக இருந்திருப்பார். ஏன்னா நான் 87 இங்கே வந்திருந்தேன். அவர் உங்களில் ஒருத்தர் இவ்வளோ பெரிய லெவலில் போயிருக்காரு. இன்றைக்கி நான் வந்து பயப்படுற அளவுக்கு, நான் அவருக்கு மரியாதை கொடுக்குற அளவுக்கு அவர் உயர்ந்து நிற்கிறார். தமிழ் எதுக்கு பேசுகிறேன்னா, மொழி ஒரு தடை அல்ல. யூ ஹாவ் டு இன்டராக்ட். We were actually begging students to raise questions in the previous session and the session yesterday. That should not happen here. You have an opportunity to talk with a top intellect, okay, and a very established professor here, and therefore I'm just presenting him to you with all kinds of happiness and satisfaction. Sir, we invite you to give your lecture. Feel free to interact with him.
So good morning everybody. So I would like to thank the principal of American College and the vice principal for giving this opportunity, and also the organizing people. So I would like to again thank your vice principal for giving this introduction, and yeah, he made a long introduction. But certain points I also would like to insist you, because in this 1 hour, because the screen is also very small, I don't know whether it is visible to you. I am not going to focus on some mechanisms, rather some maybe at least some 50 points which are very latest, like in science, in genomics, and mainly like, so what are you going to get out of this lecture is, at least you should set the goal for your career. I think as mentioned by sir, it's true that I came from a village, and while I came to MSc biology, I had huge reluctance to talk to professors, because at that time we don't know how to speak in English, and now also I don't know whether my English, I definitely, those who are like good in grammar, there will be a lot of mistakes, but now language I don't care. Okay, so we came from a village, but still we could do something. I don't say that I have reached everything. There are a lot of things to do in this country and in this area, but still we can do some science and we can contribute something. And so language is not at all a problem. Don't care about your language. And also another thing is, also don't care about the I don't know, so the people say something, somebody has a good IQ, good knowledge. I myself believe that I am really not good in I am not an intelligent person. But only thing is, I am, but it's true that I am a hardworking person. So language, intelligence, nothing is necessary. But but only thing is, we can get our share. So there are a lot of opportunities, and you all from a good college in the city, and then you all good. I am very happy that I am in front of you, and I have a lot of my friends here. Raghu is there, my junior MSc biology, Dhanjay, and then some of my colleagues are there, and sir is there, and we have a lot of known faces, and yeah. So American College, Vilathurai sir is there. So he's also did PhD with us in School of Biological Sciences, and yeah. So touch yeah. So today I will talk about some basics of human genomics, and then maybe something about what are the experimental models available, and some modern areas like epigenetics, epigenomics, personal genomics, and then biomedical genomics, the title, and personalized therapy. And also I will talk about what we are doing in the laboratory at Americancollege University, and something about the career opportunities. And you all know very well, I think I understand that you are from microbiology and zoology. I think we have artificially divided the subjects into zoology, botany, microbiology, biochemistry, but everywhere is biology. And today, we all, if you want to go for a job in to an industry, okay, and then everything is biology, everything is molecular biology, and then few disciplines. There are so many areas, so many techniques, but if you are becoming a master in one area or one few techniques, that is enough for you to get a job and then excel in that. Okay, so you all aim for a very big career, so getting a job and then settling, not necessarily, because At this stage, if you go to a company, after completing PhD, if you go to a company, you can easily get a salary of like, more than 2 lakhs or so, maybe at the age of your 45 or so. But so you aim for some very big things, or you can go abroad for your PhD. But now if it is like, but you should prepare for your GRE and everything. Or like, after doing your PhD, you try to go abroad. There are a lot of opportunities. Okay, but everywhere we don't want to like, spend money. Everywhere you will get fellowship. In India also, after PhD, for PhD itself, you can get the fellowship. So you can keep preparing for the net and other things. So in biology, basic thing is like, you have cell, you have nucleus, you have chromosome. Your chromosome has DNA. All of you know very well. And in human, we have 23 pairs of chromosomes. So that's not new to you. But our DNA is very, very long. Like if you stretch out the DNA, this is the length between like, sun and moon. So I think some 6,000 times or so. So such a long DNA is like really packed into 23 chromosomes. So our like, maybe the diameter of nucleus maybe few microns or maybe less than a micron. I don't know exactly. So within that is really packed. So you can understand that the DNA, the genetic information is highly packaged, tightly packaged in the nucleus and chromosomes. And let me skip certain things. Now, maybe one point here is you can see different organisms. We consider humans are the advanced species, but there are some. So then logically one can ask a question: which organism will have the biggest genetic material? But it is really not a human. There are some plants; they have very big genome size. So there is no direct correlation between the advancement or like complexity and the genome size, because we still believe that humans are complex systems, but some plants they have like, larger genome size. That's probably because of the ply. So they have like, more sets of chromosomes. And yeah, so plants have the largest genome size, not the mammals. Okay, so a long time back, people are talking about molecular biology. They started like, isolating the gene and studying the gene, getting the sequence of the gene. But 20 years back, people started sequencing the whole genome. So that means by spending some $10 million, and few companies and some institutions joined together, and then they sequenced the whole genome, all the DNA present in the 23 chromosomes they sequenced. So that is the genome sequence. That was a very massive effort. As I said, it costed few $1 million. Okay, but now in 2021, now if I want to sequence my own DNA, it is not going to cost more than some 10,000 rupees or 20,000 rupees. You can sequence your own DNA. Even in Madurai, for some disease diagnostics, people are sequencing their whole genome sequence. Okay, you can understand whole genome sequencing was costing maybe few $1,000, sorry, few $1,000,000 at that time for a single genome. But now the technology has become very cheaper. Like, if you spend $100 or so, you can sequence your whole genome. So the technology has really it's growing, and then it is coming for the real applications. So I was talking to sir, and then he was saying that students don't like molecular biology, and in your board of studies also students suggested don't include molecular biology, it seems. But whether we like it or not, see, everybody everywhere. So now people are using DNA for everything. You cut the DNA, paste with something, and create new protein, or create the protein which is necessary for therapy. You think about the vaccines today. Everywhere it is biology. And then this virus COVID-19 diagnosis, everywhere it is molecular biology. Okay, so we cannot just skip molecular biology. There is nothing complicated in molecular biology. So it is definitely it will create a lot of job opportunities. So now if you go to this website ncembl.org, here the whole genome sequence of few hundred organisms are readily available. For example, if you want to download the genome sequence of the humans or like mice, you can do that. It is readily available. So that's what like long time back it was a very, very costly event, and now it is openly available for us, the researchers. Okay, and then one more point which I would like to insist. In human, we have about the genes and proteins. So we have approximately 20,000 genes in the human genome. So but the biology is so complex, because I believe that the nature will not allow us to understand everything completely in science. Definitely, like we may know one part. So now, as of now, scientists know that we have 20,000 genes, but how that 20 genes, but now I am saying that the human genome sequence can be done in $1,100, and we can do anything. But can we make a cell, or can we make a cell to develop into an adult or embryo? That is not possible. Okay, so the nature is, as of now in science, we can't do anything whatever we want, but we can do certain things here and there. That is what, with that we are doing all the things. So we have 20,000 genes, but still there are a lot of small RNAs, non-coding RNAs. The system is so complex. So you all know very well in recombinant DNA, you might have studied, you can cut the DNA. That's what people used to say, cutting and voting of the DNA can be done. And so you can create new combinations, you can produce proteins. That's what, now, in diabetic people, they are using insulin. Insulin nowadays, it's because long time back, you all know that people used to take thousands of pigs, kill them, get the pancreas, and from that they used to isolate the insulin, and they used to use that for therapy. But then sometime back people were using but now what can be done is, you can now, I think you might be knowing that there are a lot of insulins, and insulin can be just synthesized in vitro by using humans own DNA. You can produce human insulin, and then that is, you don't need to kill any organism, only you have to kill E. coli bacteria, and that can be done. So any protein in our body can be modified, but not within our own body, but for therapeutic use. You can you know how to take a photocopy of a paper. You took the Xerox copy. Similarly, DNA can be copied, and in the laboratory there is a machine for that, PCR machine. And DNA also can be copied inside a bacteria. Inside a bacteria, you can put the DNA, and you can ask bacteria to replicate it. That is possible. So those are and again, between human and chimpanzee, there are a lot of similarities, 99.99% or so. So yeah, lose contact lose connection. Okay. So that means, there are a lot of DNA which is similar between chimpanzee and human, but even among us, the DNA is 99.9% similar. But still we are different. That's what the people used to say, that in a plant, no two leaves will be similar, because they have the same DNA. But still two leaves will not be similar. Similarly, we all sharing 99.9% same DNA. Okay, so even no, even father and son will not be alike, except zygotic twins. Nobody will be alike. So the reason is, we have more than 60 million regions in the genome which are polymorphic, which are there may be a single base which may be different between one individual and another individual. Likewise, we have 60 million regions in the genome, in our genetic material. So that makes us different. That will be there in different combinations. Okay, these are nature's wonder. So it creates all our same species, they interbreed, but still the DNA is different in 60 million regions. So that makes us different from each other. Okay, and just we are not clones. Now in biomedical genomics, people are trying to use this as a advantage. Okay, now individuals are different. Now what is the problem in medicine today? You take any drug, the doctor is going to give the same drug for everybody, but the same drug is not going to work for all the 100% of the people. It may work in 50% of the individuals, or 60% of individual. In another 40% it will not work. That is a problem in therapy now, or maybe it may not work efficiently. But in future, we want to attain a drug has to, if a doctor is going to give a drug, it has to work. That is the ultimate aim. So why it is not working? One now, it may be for different biological reasons, but now can we use these kind of now I told 60 million polymorphic regions are there in the human genome. Can we use this as a diagnostic marker? But people have done something like that. Why we are getting this kind of ideas is, people have already done that. There are some drugs, people get the DNA sequence, and if it is a particular DNA sequence, a particular drug will not work in that individual. There are some examples about the drug. You can go and search about the drug warfarin, and if that will be very lethal to some individuals. So before giving the drug, people used to sequence the DNA. So that is a successful model. But now for every disease, can we use this kind of DNA sequences linked to a drug response? That is one major area in biomedical research. So as I told, now the genome sequence is nothing. It is openly available. Okay, if I am going to sequence, if somebody is going to sequence my own whole genome sequence, they give me it may be few terabytes of data. If they give it in a pen drive, what can I do with that? It will be just like ATGC, ATGC. It will go like that. I can't interpret that. So in biology, the problem is the genome sequence is already available. The genome can be easily sequenced. But how to understand in terms of biology, how to understand in terms of function is the challenge. Because though the genome sequence is available, but we want to understand the functions. For example, genome can be easily sequenced. That's not a problem. It's costing only $100. But how from a single zygote, all the different cells, for example, we have nerve cells, we have muscle cells, and different gonad cells, and then red blood cells, white blood cells, nerve cells, and different types of cells we have. We have different organs, but the genetic material is same in all the cells, in all the tissues. But how the cells are being different? So that is called the area is functional genomics. So you have to understand the functionality of the genetic material, but that is very hard. So that's what sometime back I told you, like yeah. So we have only 20,000 genes, but still they are like tightly regulated, tightly controlled. So no two cells will be similar again. Sometime back I told you, no two leaves in a plant would be similar. Similarly, no two cells in a body also would be similar, because everything makes the environment. That's what one of my teachers used to say, that imagine there are two like twins. Okay, so in the very early stage, you leave one in like somewhere Tallahulam, and leave another person in the Harvard. So obviously the environment would make a lot of differences. Okay, because a lot of things we are picking up from the environment, not only like biological things and social things, and so many learnings are coming from environment. So similarly, cells are also like surrounded by so many some micro environment. So that determines cells like function, features, and everything. So though all the cells have the same DNA, but the cells are not similar. For example, eye cells are different from nerve cells, so they have different functions. So another area is genes are tightly regulated. You know the structure of the genes. You might have studied the lac operon and other things. So you have the genes have coding regions, genes have a regulatory region, and this is like a switch and light. If you turn it on, a light will be glowing. And similarly, in the promoter is tightly regulated. So the complexity in biology is not the DNA sequence. DNA sequence is cheap now. If, for example, if I want to sequence my own DNA, within a week I can sequence by just sending the DNA to some companies. And but the challenge is how to understand the biology is different. And one of the complexities is regulation. The promoters are regulating the gene expression, because now we have 20,000 genes approximately, and 20,000 genes are differentially regulated. In lung cells, maybe 100,000 genes are expressed. In liver cells, some other 1,000 genes are expressed. But the system knows which is liver cell, which is lung cell. Okay, so that's is a brilliant system. And yeah, but sometimes what happens is in this kind of in genes, some problems are happening that we call mutations, and so it causes a disease. Sometimes some problems are happening in the switch or in the promoter region, and so gene regulation is getting affected. That also causes some disease. There are some examples, but now the point is, for example, in cancer cells, what happens is, for example, in this case, this M stands for methylation. Though I will talk about that methylation later. So the DNA is like methylated in the promoter region. If it is methylated, then the DNA cannot be turned on. The gene expression cannot be turned on. So for example, in some examples in cancers, it is the promoter is methylated, and so a good gene which is necessary for our benefit is not expressed, and that causes cancer. But now what people have identified is, people have identified some chemicals, drugs, which can remove this methylation, and so bring back to the normalcy. Okay, so why we are studying all these things is, first thing is to understand the biology. Second thing is to use it for an application. Okay, so I always have to see the things, whatever I do. See, what I am doing is, I am doing my career development. Okay, whatever but of course during the process my students are also get benefited. I'm not going into the lab, my students are doing all the work. So in the process they also will get benefited. Okay, and similarly, whatever we do in biology, there must be an application. Okay, so yeah. So you guys ask a question every day at the end of a day, what I'm going to get from this lecture. Okay, what I'm going to get from this course today, what I'm going to get from this paper. If it is not worth, don't do that. Okay, so everything you have to own it. I'm not that cunning person to say all these things, but the thing is, we should get something. Just understanding biology doesn't matter. Who cares? Unless and otherwise, if you can convert this into a medicine or into is an application, there is no point in that. So maybe I'll skip certain things. In the laboratory, you can understand the gene expression by PCR. So those kind of experiments can be done in the laboratory itself, and the proteins can be expression can be identified by Western blotting. Let us skip those things. And nowadays there is a very good method by which the proteins can be visualized in the cells. There is a fantastic method called fluorescent microscopy or confocal microscopy, where inside the cells you can see here there are beautiful cells, and then they are differently. There are a lot of fluorochromes. Okay, and there are thousands of fluorochromes are available, and they can be specifically used to track the mitochondria, nucleus, cell membrane, and so on. So these are the techniques which can be done. Actually, normally for our MSc program, we used to do these practicals, like how to track the stain the cells colorfully. Okay, and sometime back, you have seen that we can do Western blot, we can do RT-PCR, we can analyze one gene at a time. But in genomics is a field where you study everything. Like don't look normally in molecular biology, people will be studying one gene at a time. But in genomics, you'll be studying all the 20,000 genes at a time, because now in biology it is a genomics era. Okay, and then again, as I said earlier, one of my views is we don't need any brain for doing science. So now science is like just having money and doing some technology. That's what, like if you go to big conference, somebody will be taking the donkey, because nobody has done the whole genome sequencing for donkey so far. So then somebody will be presenting, I have sequenced the whole genome sequence of donkey, and this has this much of homology with monkey and all those things. So now in India we have a lot of indigenous plants and so things. People are doing that. So do we need a brain for that? No, not necessary. Only thing is we need interest. We need, something we have to make meaningful out of that. And so nowadays everything is the whole in biology. In my view, everything is sequencing the whole genome. So people are not doing the molecular biology. Molecular biology is like very focused for one gene. People just sequence everything, and from that you can understand the expression of all the genes at a time. For example, in cancers, people take a small piece of tissue, isolate RNA. From that they will do RNA sequencing. Now all the 20,000 genes expression in a single patient will be known in 1 or 2 days. So that will give huge information. That is called microarray studies or RNA-seq studies. And but biology is very complex. I think I am also reiterating this point again and again. Biology is not that simple as we think, because we have 20,000 genes. But when the genes are expressed, where they are expressed, still we do not know completely. We are trying to understand to some extent. And complexity is very high, because 20,000 genes is very small number considering our complexity. But 20,000 genes, in which cells they are expressed, at what time point they are expressed, and actually these genes are responsible for diseases also. Because if there is something wrong in the disease gene expression, or if it is wrongly expressed in wrong tissue, for example, if a protein which needs to be expressed in a liver, if it is expressed in a lungs, that will create a problem. So those kind of problems are there. And another complexity is, it's not like everything I'm talking so far about DNA and RNA gene expression, but whether the RNA is converted into protein is a problem, whether the protein is expressed or not is a problem. And then proteins are also undergoing a lot of post-translational modifications. Okay, so maybe let me skip that to avoid the complexity. And then we have 20,000 genes, but there are a lot of crosstalk among the proteins. Maybe in a classroom you may have 20 students, okay, or forget about the students. In a staff room we may have 10 teachers, but they may have 25 groups. That's possible, no? In different time points, in different combinations they will join. Similarly, we have 20,000 proteins, but they join together and then they do a function in different combinations. That is also the complexity. So that's what I'm repeatedly telling. We have 20,000 genes. That is a small number, but still their combination makes a phenotype, different cell phenotype, different functions. And then there are different mouse models. In some experiments people use chimpanzees, and then zebrafish is a good model, and Drosophila is a model. In genetics you might have understood a lot from Drosophila, and C. elegans is a model, mouse is a model. For example, one good thing about C. elegans is a model. I know some colleagues in Alagappa University, they are working on C. elegans. It has very defined I think we can't say how many cells we have in our body, but C. elegans has 959 cells, and the people can understand the developmental biology so easily. because they can track everything. They know which cell is going to become which organoid. So everything they know. So coelicans is a good model in that aspect. And zebrafish is a small fish, and for example, if I want to screen 1,000 drugs in the laboratory, in a small well plate, in each well I can put a zebrafish, and then each well I can put one drug, and then try to see which drug is really beneficial. Those kind of things can be done with zebrafish model. So each model has its own advantage, and mouse models are authentic models because they are much closer to human. And in our laboratory we use cell lines as a model, because people have established a lot of cell lines. Cell lines means, for example, if a person has a breast cancer, from the dissected out breast tissue, people take some cells and then they maintain as a because cell lines will continuously grow in the laboratory. You can grow for hundreds of years in the laboratory, and they can be cultured in the laboratory. You can freeze, if you go for a vacation, you can freeze the cells, you can come back, revive the cells, and then you can use them. And you can turn on the gene expression, turn off the gene expression, anything you want, manipulation, it can be done in the cell lines. So cell lines are also good models, because there are more than 2,000 cell lines of lungs, liver, brain, organ are available already. And another area is, so any questions so far to ask? I think we can have this as an interaction, and then another area, yeah. No, because in zygotic twins, the DNA would be 100% similar, but only the environment might be making some differences. Same environment means still the micro environment. For example, imagine you and another student are same in the same place, but still maybe the visual may be different here, and micro differences would be there. That might be making some differences. DNA is same, but now the concept in biology is now I am repeatedly telling, we have 20,000 genes, we have a genome, and that alone is not determining everything. I have deleted the slides, but since there are some microbiology students, now what people are saying is, we have 10 times more number of microbes in our own body. You see any organ in our body, external organs, mouth or whatever, everywhere we have bacteria. But these bacteria are, I'm not talking about the harmful bacteria or pathogenic bacteria. We have lot of bacteria, they are helping us. They are maybe, I'm not talking about probiotics. So we have our gut microflora. There are a lot of bacteria which is probably helping us. But that bacteria might be varying between you and me. Okay, if I am taking alcohol, then some bacteria might not be there in my system. Okay, if I say, imagine now we all using, from morning onwards we are using chemicals. Okay, I'm not against the chemicals, because without chemicals we can't live now. From starting from toothpaste, we are living with chemicals. You think about all the chemicals. So now the chemicals might be influencing the bacteria also. I think people have done one experiment. I don't know about your college. Many people, they used to wash their hands before. I think all of us used to wash our hands before we take our lunch. Okay, and some people, they might be using soap. And people found in one experiment, those who are using soaps regularly before their lunch, they found that, because obviously soap will be entering the body, and that would be killing some of the bacteria. So then obviously that would be, this is not going to kill the individual, that is not going to create a big disease, but still microflora of the body might be different between different individuals. Yeah. So those who are using plain water, and those who are using soap, and then obviously soap might be, then the question is, what we need to know is, we have so many soaps in the market. So which soap is so now you can understand there are differences. So all the things are not coded only in our genome. The bacteria present in our body also make. I think another example is the childbirth by normal mode and then cesarean. And people found that during the normal delivery, some specific bacteria are getting exposed. And then think about feeding habits, everything. So all of our habits are going to change our microflora. So those also might be making some changes. So the bacteria present in our body, which is not related to our genetics, but that is also influencing the health. That's what you can see. The aging is different in different countries, in different pattern. That's probably because of, not only because of lifestyle, food habits, and also probably because of the bacteria. So in DNA we have everything. DNA code is there, ATGC. But above DNA we have DNA methylation, and that also determines, because people thought that whole our the DNA is our blueprint, it has everything, the ATGs is the blueprint, but now people are giving much importance. Above the DNA you have the methylation, so that also decides that whether a particular, see, gene is same, gene sequence is same, but whether the gene has to be expressed in a particular tissue in an individual or not, that may be decided by the epigenetic factors also. There is the DNA methylation, and then and also histone modifications. The DNA is wound around the histone proteins. You might be knowing that. You know, is a nucleosome is there. DNA is wound around the histones. So the modifications in the histones also decide the gene expression. So that is also part of epigenetics. And there are some concepts. You are what your mother ate. You are what your grandparent ate. Because what people are talking is, while some people have seen some examples. If the mother is smoking while pregnant, that is going to affect the, not affect, that is going to make some epigenetic modifications in the offspring. Not only in the offspring, in the gonadal cells of the embryo. So from one generation the information can pass on to the next generation. This is not genetics, directly DNA change, but epigenetic marks are changing, transmitting to the generation, and that is another concept which is emerging. Let me skip. So maybe I'll try to explain this. For example, in sperm and egg, you have lot of DNA methylation, but while it becomes blastocyst, many of the epigenetic marks, DNA methylation and other things are coming down. Then subsequently, like in the progenitor embryonic cells, you are again losing them. The only DNA remains. The epigenetic marks are not remaining. But eventually, while it becomes adult, again the epigenetic marks are coming back. So there is something else, So far people are sequencing only the human DNA, but the marks on the DNA are also important. That is one area, epigenetics and epigenomics. Let me skip. Okay. Now the major topic what we are talking today is the biomedical genomics or genomic medicine. How you can use the genomics knowledge or genome sequence knowledge for therapy, for medical applications. And see, in about sometime back I told you, if you give a drug, it is not going to work for all the individuals. It may work, for example, in a group of individual, one drug may not work for individual, or it may work for individual, but we have to identify. How are you going to identify? So only by doing a lot of trials, only by understanding the genome, we can do that. So that is the power of genomic medicine or personal genomics. So this is a cartoon representation. You have different individuals with different colors, but you have to segregate them. That is called personalized medicine or stratified medicine. Okay, don't give the same drug for everybody. Try to group the patients into different categories, and so that is a way. That is a way in which the genomic science is moving. It's not like one drug going to work for everybody. That's what we have seen in the COVID-19 situation also. Some people are less symptomatic. Some people are severely symptomatic, and then some are hospitalized. It's the same virus, but still it varies. I think age difference is something else. So in any disease, if you are going to segregate the population into different subsets based on their vulnerability or based on their response to a drug, that is the future of genomic medicine. So you all youngsters, if you get trained in one or the other areas, you can contribute in one or the other way for the development of personalized medicine. As of now, personalized medicine is in the early stage. It is already being in use for some cancers and other things. But in future, people are going to explore this more. They are going to link DNA with everything. Yeah, already I told you that we have 60 million regions in the genome. We have sometime back I told you, $100 million was spent for sequencing one genome. But 5 years back it has become much cheaper. Then people started doing 1,000 genome projects. 1,000 genome projects means in a country you sequence 1,000 individuals, their whole genome is sequenced, and then try to understand. For example, in India, 1,000 people. Recently, there is a work going on Genome Asia. From different Asian countries, they are sequencing, I think, 1 million individuals or 10,000, I don't remember, and then we can analyze them. But now the point is, this is not somebody doing somewhere, okay, and all this data is openly available. Now, if our MSc programs are rightly designed, okay, in my view, if I am a student in right now, I will be giving more importance to computational languages. Okay, now if you go to the laboratory and isolating DNA and doing all those things, in my view, those things are merely technician jobs. Anybody can do that. Nowadays, even there are machines came actually. In COVID-19, for RNA isolation, people are using machines. If you just keep the blood, RNA will come out. Okay, so isolating DNA, molecular biological techniques are not skill oriented. But what is really important is analyzing the data. Now the genome sequence is 1 terabyte of data. How are you going to analyze? As a biologist, in biology, you might be seeing the chromatography and all gel and all those things, but it is very easy to see. But 1 terabyte of data, how are you going to visualize? So for that, we definitely need some computational knowledge or computational. I think there are tools available, but we don't need to transform ourselves into computer science or IT person. But at least we should be able to use some small codes, and we should be able to. That's what. Well, I was a postdoc in Singapore, and in that laboratory, those who are working in the laboratory, they would be working. But whenever the papers are published, the first author would be the person who would be analyzing computationally. So now also people are doing, see, there are two ways. You can see that now in elections, long time back, people will be doing all the canvas in the streets. But now people are sitting somewhere, so they are having the IT wing. They are managing everything now. So they know everything. So similarly, the data also working laboratory is one part, but computationally controlling and analyzing all the data is big, because I told you many times, $100 with 10,000 rupees I can sequence my own DNA. But if I want to analyze, 10,000 rupees is not enough. So analysis is very costly. So people still the world needs lot of persons who can understand the biology and who can also do some computational analysis. Now imagine a genome essay is 100K project, 100,000 genomes. So that data is openly available. So in our laboratory, we are downloading the data and then we are analyzing that. But we definitely need the help from programmers and then bioinformaticians for that. But many times we don't need to go and study bioinformatics. Just learn one language, maybe R or Perl, Python, and then you can own that. See, if you are a computational person, if you are a biology person, and if you know a little bit of computational knowledge, then you can own the data. Because otherwise, for biologists, this data is, how do we visualize the data? If I sequence my own, long time back, one of my colleagues, what he used to do is, whenever they are analyzing the DNA sequence, he will print out everything and then he will keep and then align. But now computationally, you have to see, so imagine there are 100,000 genomes. Each genome sequence is 3.1 10 power 9 base pairs. With the naked eye, with the screen, we can't see anything. But unless and otherwise we have computational view, computational handling of this data, that's hard. Now in COVID-19, the data is available. So far, people have sequenced almost a million viral samples. You can take the data, analyze, and then make a publication, and all those things are possible. But we need some computational skills. And another area is since people are doing the 1,000 genome project all over the world, is we can understand the migration. So how we got into this place, where did we come from Africa, or in through which route we came, and all those things, because that data is now slowly the data is available for so many samples, and those kind of migration pattern can be done. But it's not, for example, in many countries people are interested. In Singapore, they used to do a TV show, because mostly in Singapore there are a lot of Indians are there, and they want to know that from which place they came, and they do some TV show like that. But similarly, we can do at the continent level, since from the DNA sequence. Then another area is personal genomics. Now I'm sure that in next 10 years, for many of us would be having our own DNA sequence, which is possible. And it is also in some countries, the government itself is initiating personal genome project. So in that, what imagine you want to sequence the whole DNA, you just even now there are some companies, you just order and they pay, and then they will send a kit, and you just spit, and then send your saliva to them, and they will isolate. DNA can be easily isolated from buccal wash, and the DNA can be isolated, and then they will create a login password for you, and they will put your whole genome sequence. But they, from that, what are the disease for which you might be susceptible, what are the drugs for which you would be responsive, what are the drugs which would be not responsive, and all the data from that it can be done. But as of now, even we don't have that much biology. The genome sequencing is possible, but understanding the disease susceptibility still is in a early stage, and so it is not useful right now, but in future this will be really meaningful. Okay, so in simple point of view, biomedical genomics, like what people are doing is in diabetes they are taking 10,000 individuals, diabetic, non-diabetic, sequence the whole genome, trying to understand what all are the variations, then how that can be used for therapy, how to, because now diabetes is considered as a single disease, but in reality diabetes might not be a single disease. I'm talking about type 2 diabetes itself may be categorized into 10 subtypes. Then for each subtype, if you give different drugs, then it may be the best possibility, and people are working on that. Okay, these all are the areas in which we are working in our laboratory. One is cancer genomics, so we are trying to understand the genomes of tumors, and we have collaboration with Mother Meenakshi Mission Hospital and doing a project in gastric cancer. We sequenced some 50 individuals whole tumor, and then we are working on that, and our aim is to develop a targeted therapy. And similarly we are working on also, like oral, breast and liver cancers. And another one is metabolomics, and in biochemistry you might have analyzed glucose or lipid or something, but now the whole metabolome can be studied. Whole metabolome means from blood you can study hundreds of metabolites or like biomolecules in a single. There is a machine available for like GCMSMS, gas chromatography, mass spec. You can analyze every, just from 10 microliter of serum you can analyze everything, and that is called the metabolomics, because metabolomics would reveal like disease biology and everything, and nutrigenomics in one area, and drug mechanisms. So in we are growing a lot of cell lines in the laboratory, and from that we are trying to understand the mechanisms of the drugs, because we are using many drugs already, but still we do not know the mechanisms completely. So we are trying to understand the mechanism and repurposing. Sir was also saying some time back, because so far we talk about the side effect of the drugs, but we have the habit like நமக்கு எது ரொம்ப நல்லது பண்ணுதோ அதை தான் திட்டுவோம் இல்லையா, காலேஜை போய் திட்டுவோம், ஆனால் 10 வருஷம் கழிச்சு காலேஜ் அப்படி இப்படின்னு பொழுதுட்டு இருப்போம். ஸோ அது மாதிரி வந்து ட்ரக்ஸை வந்து எல்லாரும் வந்து அவ்வளோ சைட் எஃபெக்ட் அதுன்னு சொல்கிறோம், பட் ஆனால் வி ஆர் லிவிங் ஒன்லி பிகாஸ் ஆஃப் த ட்ரக்ஸ். பீப்புள் யூஸ் டு சே தட் ஒன்லி பிகாஸ் ஆஃப் த ஆஸ்பிரின் the life span of the individuals got increased by 10 years அப்படின்னு சொல்லிட்டு there used to be a statistics. So now in all the houses I'm sure that there is a place for medicine, either your parents or grandparents used to take medicine. Okay, so similarly so the best thing is the ideal thing should be, now we used to thank sun by pongal no. So we are thanking sun for giving everything sunlight, and then so all the agricultural foods. Similarly we should have a day for medicine. We should keep all the medicine and then do a prayer. So medicines are playing that big sometime back, like heart attack used to be like deadly disease. Now there are a lot of evidences, people like after heart attack they are taking the drugs, blood thinner and everything, living happily. So drugs are playing a very big role, and we are living because of the drugs, and not we, like at least I'm talking about the society, and we are really getting benefited. But obviously there are some side effects, and then eventually by doing some careful experiments, careful design, we have to try to bring down the side effects. And repurposing is sometime back, one story I used to say very often. In US they found some group of individuals, those who are taking aspirin for headache. Sometimes if you have a headache, then you will take an aspirin. Some individuals, they used to get a headache periodically, maybe once in a month or so, or once in 2 weeks they used to get a headache, so they just take an aspirin and then it goes. And later people found that the group of people, those who had aspirin, the statistics of them getting colon cancer was less compared to the people those who are not having headache, and so they are not taking aspirin. Okay, so from that they found that aspirin has the capability to bring down some tumor suppressor role for colon cancer. Similarly there are a lot of examples. This is not a very big story. And similarly people found some anesthesia which was in breast cancer therapy. Later they found that that is anesthesia, still that had some effect, anti-cancer effect. So the drugs can be repurposed for some other applications. We always talk about the side effect of the drugs, but the drugs can be repurposed for other applications. So we are working on taking the existing drugs and then against cancer. Recently we have published a paper, and one antibiotic has very good effect against some pathways in gastric cancer. And we in India we have a lot of medicines, Siddha medicines, Ayurveda medicines, and then homeopathy also used, but we don't know the mechanism. And also we have a lot of home remedies, but we don't know the mechanisms. But we are working to understand the mechanisms by using genomics approach. And recently we are also into the the Keeledy DNA project. So there are a lot of bones from archaeology department, from human and animal bones, and also some plant remains. So we are establishing a laboratory and we are sequencing the ancient DNA and trying to understand the migration history, everything from that. And similarly, recently we are also into project we say that yoga has some beneficial effect. I think many of the teachers might agree. It is because, but still we don't know the molecular mechanisms. So we just started a project at MKU. So before yoga, just take some blood, and after yoga, take some blood after 45 days, and then what kind of factors, stress factors changing. We are working on that. Yeah, we spread it out into originally, I was working on cancer only for the past 10 years, but recently in the last 1 year, we just really changed into so many other things also. Okay, it's already 1 o'clock, maybe your lunch time. So maybe just in next 5 minutes, I'll try to close everything. So we are culturing a lot of cell lines in the laboratory, and then use them for various purpose. And by using various methods, we are trying to understand the whole genome-wide variations. And this is one of the major logics with which we are working in gastric cancer with Meenakshi Mission Hospital. So what, see, as of now, gastric cancer is stomach cancer. Stomach cancer, only some 2 subtypes are known in clinic. Clinicians subdivide into 2 or 3 subtypes. But by using genomics method, we are trying to subdivide into 8 subtypes. So we have done that. Now the point is, for 8 subtypes, you can have 8 different drugs. So in cell lines, we also made a drug screening system, and we have drugs. So now we are trying to make this as a concept, and then you try to diagnose while a patient comes to the hospital, you try to diagnose what is the exact subtype, and so rather than giving the common drug, you try to go for a specific drug. But this is a long way, because anything translating from laboratory to clinic is not that easy. It has to go through a lot of regulatory clearances and everything. So we are working on that. That is ultimate aim. So now you can see here, these are tumors. In some individuals, some pathways are highly activated, so you give a specific drug. In some other individuals, some other signaling pathways or cellular processes are dysregulated, then you target them. You can differentially target different pathways or genes in different individuals rather than giving a drug, same drug. So this is what I repeatedly say: personalized medicine, genomic medicine, or targeted medicine, or stratified medicine. So we have a screening system. We have established some cell line in the laboratory. So that's what we do is we take the cell line and screen in. You might have seen 96 well ELISA plates. In each well we put the cells, and then each well you put one drug. So we are screening thousands of drugs, and so in a single go you will understand the mechanism of hundreds of drugs. So this is a screening. From that you can identify some drugs which are like really inhibiting the pathway very well, and so we identify the drugs. So drug screening is one of our areas we do. I think one of the homeopathy medicines, ornithagalam, because nobody knows the exact mechanisms, we found that it really inhibits some pathways. And now again, as shown here in the red box, the ornithagalam is inhibiting these pathways. And so don't give this drug for all the patients. You give this drug only for the patients in which the pathway is activated. Yeah, we have done a small cohort from Madurai, and then we are screening thousands of drugs. So we definitely need some good students. If you clear some net or something, if you have strong interest for research, and if you are interested in 1 or 2 in a vacation 1 or 2 weeks, you come. And again my point is not for coming and learning. Come and do the works, and then let you also get the benefit, and the lab also get the benefit, and we can do that. So we are understanding the mechanisms of the drugs. Let me stop here, and then see again. Now, sometime back I told you from Madurai we have sequenced the 50 individuals stomach tumor. Okay, but already in the open access we have the sequence of 11,000 tumors. So the data is already available. But the only thing is, we have to analyze. So again, computational biology is very important. Reanalyze, make models, hypotheses. You can make a publication, or you can make and to test in the laboratory. So this is a number. So this kind of data is available in any field. The data is available. The data is openly available. Okay. So in carriers, there are a lot of. So I wish this is one area. like bioinformatics, you can't go and study because you are studying biology and then this one, but just learn some coding languages, some basics, and then subsequently you can sharpen that. Anything is possible in the world if you are interested, and if you put some small efforts. You don't need to work very hard. You don't need to change upside down. Imagine you are learning a language, one language in 1 year, another language in next year. Somewhere it would be helpful. So coding, because nowadays even the LKG students are learning coding, and then there are some some system available for them. This is just some statistics. You can get some. We have a programmer. Say all the research fellows, they are getting 16,000 rupees salary, or 30. Even if you clear net 33,000. But if you are a programmer, you will get 40-50,000 rupees even in our own place. So recently we are into the ancient DNA. So we are collecting the bone from Kilidi, and there are so many places, and from animals also. We can try to understand the migration pattern, and we are working with Tamil Nadu government's archaeology department. And the plan is, as I said, we have the genome sequence of thousands of individuals from all over the world. So now, while we have the sequence from Kiladi, Kondahai, and then from Sivagalai, so we can try to match that with world population, and then try to. So we are working on that. And again, also, like people were using a lot. You might have seen a lot of offering pots in all the archaeology places. So we are just scraping them, and then the logic is, so whatever the food materials they may have used, by using some GCMS or LCMS, we can try to find out probably what kind of food materials they have used. Also by sequencing the DNA from that, we can try to see what kind of DNA they have, what kind of food material they were using at that time. So all those things we can identify. So we are expanding that project in so many directions. So there are a lot of possibilities, there are a lot of job opportunities, but you dream about that, you work on that from now onwards, because whatever your MSc degree is, see, college will, for sure, it will give MSc degree to you. If you come to college every day, even if you say that I don't want degree, college will give the degree to you. But you have to plan what are you going to do. But with that degree, you can't do anything. Okay. So only thing is you have to sharpen yourself. You have to keep observing the surrounding. You have to keep looking for the job opportunities. Anything is possible in this world, but you work on that continuous manner. But that nobody would teach you. Only you have to, like, in any way, all of us are going to live in one or the other way. But still, like, see, you all in a good college, you all surrounded by good faculty members, good atmosphere, and then, but you all should excel well. So that is the thing which is necessary. Okay. And if you do PhD in India, you would get a fellowship of 33,000 as of now. So for that you have to clear the net. So I would say that don't rather than studying for your see, yeah, our MSc students, but not I don't see that kind of atmosphere now. Some 5-6 years back, some students, they used to come to class regularly. They will ask doubts. They will answer everything. They will get even first mark. But after completing MSc, they may not get into their next phase immediately. But some students, they will not come to the class at all. For the whole MSc, I might not have seen them at all. But they will write the exam, they will get 55 marks. So what they were doing was, like, they were in the hostel, they were preparing for their GRE exam. So immediately after completing MSc, in the fourth semester itself, they will get a position for PhD abroad. Okay. So nowadays the formal education is one part. You have to keep working on, because this is kind of, I would say, I don't know, this is like a living fossil system. So I am a teacher, I have to teach, you have to listen. Yeah. So no other go. இப்போ நீங்கள் போக முடியாது. போனால் வந்து அநேகமாக உங்களுக்கு சொல்லிடுவாங்க, உங்கள் ஹெச்ஓடிட்ட கொண்டு போயிடுவாங்க, இல்லை பிரின்ஸிபல் அழைச்சி கொண்டு போயிடுவாங்க. வேறு வழி இல்லாமல் உட்காந்துருக்கோம். ஸோ இதே மாதிரி தான் கிளாஸில் இல்லையா? பட் அது வேறு சிஸ்டம். பட் லைஃபுக்கு அது பிரயோஜனமாக இருக்குமான்னு தெரியல. சம்திங் தட் டிபெண்ட்ஸ் ஆன் அவர் ஓன் டெஸ்ட். நமக்கு ஒன்றும் thirst இல்லை ஒன்று அப்படின்னா அது வேறு விஷயம். பட் இஃப் யூ ஹேவ் thirst, you have to do something. There are tons of possibilities are there. So explore, excel, all the best. If you want to do research, you prepare for net. Once you clear net, you can get your PhD anywhere in India. With fellowship, you can do all the things. So there are plenty of opportunities. Still you have time. I wish all the very best. So I'll be happy to answer for any questions. Thank you, sir.
Priya gave me a title for this hour. She called me moderator. Okay. So I'm supposed to moderate the program, and there's no need for me to actually intervene. This is because he had given a handout. I'm sure if you can get a printout of it from the assistant coordinator. Okay, so you can read it yourself. Now in that, and I see a statement as Sir concludes, the current focus of pharmacogenomic research is to explore the effect of inter-individual genetic differences, on the pharmacokinetics, pharmacodynamics, efficacy, and safety of drug treatments. So this is where his engagement is. So every bit of word is something which calls for deeper insights and reflections. I was so glad that one of the friend who was seated there was asking a question, interrupting. He was asking a question, but interestingly, just about the time, the slide on epigenetics was flashed. Had he waited for 2 more minutes, he would have got the answer. So he has actually explained that very clearly and nicely. So as the speaker was trying to point out, the 23,000 different genes may be sitting there, and the 60 how many? I have to go to 60 million polymorphic regions may be there, and there could be a countless number of biomarkers there. But all these things means what to us? So you have to put it all together and have a holistic idea. And this is where you have a scientist sitting here in blood and flesh. Okay, and he is a person who's I liked him. He was actually just trying to make a comment that what is the point of you coming to my lab? For general learning? No, there should be a purpose. So I think he is the scientist of the day. So he is valuing his time. He values the time of his students. He values the time of the funds that he receives from the funding agency. That is why he is so successful. I'm sure you should join scientists like him. But for that, you simply cannot go and waste his time or knock his door without that net or a suitable clearance. So your degree that you are going to earn from here is not going to take you anywhere. Know it from us. We are teaching you and we are giving you the degree. This degree is not going to give you anything unless and until you clear your net. There cannot be any future for you in biology. But once you clear that, a world of opportunity is going to be open. And when I think Tadej joined a little later than me, when we were here, we used to see RJ, a colleague of Watson. Okay, he was actually working in the laboratory, Dr. R.J. Raman. So we used to look at him with awe. So we were actually lamenting that kind of a professors are gone. JJ, RJ, SK, these are maybe names to him, but we have seen them in blood and flesh. Okay, so we were actually lamenting and worrying that that kind of a legacy is gone from MKU, and we were very sorry about it. I think the old timers here were actually not satisfied with the kind of performance of the university, but he brought me happiness, a whiff of fresh air into us, and that legacy is not gone. He is still there. So you see a RJ here, and you don't have to travel to places, go to Singapore or US. Get the initial training here, and he's going to shape you. He's going to shape you, and he means business. He means business. He's not a crude man, but he does not want all this kind of philanthropic feel-good factors. You get into his lab, learn the technique, give him the data, and you will definitely reach your place. That is where he is forthright in being very honest. At the same time, he says that system is bigger than what is actually projected here. So Sanjay is actually teaching system biology here in plants. Okay, a system behavior you have to understand. So we had a good session, but I think we'll have a couple of questions. With that, I think the talk can be complete, and that is where you would see that the professor will be respected. Okay, and now the topic is open for discussion. I take a question from here and there at least. Come on, you have asked once, so give a chance to your friend. You can tell your question to your neighbor. This side, that side you don't have to. Johnson is an old timer, gone. He has no chance. We want youngsters or a non-biologist to raise a question. Yeah, she is Dr. Amuda, one of our thinking teachers, and she had been working for Satyabama University for long, and he is now in mathematics. She has a question. You can answer that.
Sir, very good session, sir. So I am a layman of molecular biology and all, and I have a question. So you told me that every day we live with the drugs, and
Oh, that's the mic.
yeah, and I know that 2016-17, one professor proved that diabetes is not a disease. We should not take a drugs. Whether it is accepted or not, I don't know. And but nowadays, if any lab or any doctors identify that if I'm having a diabetes and still on that, I have to take a drugs. And after that, all the disease will come to me. Am I right, sir? So there is no solutions on it. As a layman, I'm asking you. You told me that it is aspirin gives a lifespan of a person. If I have any problem, if the chance to take aspirin, then it will gives the lifespan of the time, right? In that way, I'm asking you whether taking drugs for the diabetics is advisable or not.
Yeah, thank you, ma'am. Actually, you told diabetes is a disease or not. Definitely, it is a disease, and it's a lifestyle. Nowadays, like is a relevant point which I have not touched is we are talking about genetics, but mostly many of the diseases, including diabetes, are mostly nowadays it's become a lifestyle disease. There is not mainly not because of our genetics, but mostly because of our lifestyle and environment. So it is definitely a disease. It's a metabolic disease. We have to take it. Nothing wrong in that. But see, because of that, we are getting additional diseases also. But no other go, no. Because see, in cancer, there is a concept. Some drugs are very toxic. Still, people are allowed to take the drug because that drug will improve their life expectancy. But otherwise, they will die. So it is a crude word. I should not use that. There is no solution. But the thing is, obviously, lifestyle modifications and everything might definitely help. I think it is possible to change lifestyle and then live with better metabolism, metabolic changes, lifestyle changes. Then minimize drugs. It is possible. But diabetes, I think probably it is hitting low metabolic individuals, those who have fond of foods and everything. So then it keeps the both are going in a parallel way. So nothing wrong in taking, there is no other way drugs should be taken. And another point, you are talking about the diabetic drugs. See, we are also working under a lot of evidences. The metformin, it has lot of better things, and even it has anti-cancer features. Another drug, some of you might be taking also, statins for hypercholesteremia, and people have found that it has anti-tumor effect. So it is possible, but we don't have, I don't know exactly, but definitely many of these drugs are having some better effects also. So there is no doubt about that. But side effects should be there. Definitely better effects are there. So if somebody is diabetic taking metformin, be happy. Okay. And so it may have, see, obviously it also has side effects. I think it creates gastrointestinal reflux disease, and there are certain bad things also. But see, everything will have some good and bad sides. So you can search Google better effects of metformin. You will see a lot of things. So some.
The general question. Normally, we take drugs to quell the problem. That is something wrong. Now, metformin is a safe bet. I'll tell you, but you have to take metformin along with another drug, or which will actually take care of the side effects. Metformin, as such, don't stop. It's a personal question. So, that's where he's talking about the repurposing of the drugs. So he says that aspirin, I never expected aspirin. We used to that easily consume, and we never used to have. We were students at NCC and Loyola. We had other inventions, other benefits of aspirin. Okay, so we cannot be saying it in public now. Okay, but we were warned of heart attacks. So don't take aspirin because you're likely to get a heart attack. But now he comes and tells us that it's anti-carcinogenic. And sir has actually guided us that not every drug is a bad drug. That's where this kind of the genome sequencing becomes important. What was very expensive then, thanks to COVID. Otherwise, we would not have understood the molecule.
Yeah, many of these are mathematical models, ma'am. Actually, computational models are mathematical models. So students should learn statistics and mathematics also. Other questions?
Yeah, that's it. There's a question there. Is there a question? Operate panel okay, Vidya.
Yeah, please, sir.
Gideon, check. Yeah, here is a friend. Introduce yourself. What's your name, ma? Okay, Amir. So otherwise, we will be speaking with the course. So Amir, okay, come up with the question.
So you told that it will be possible for everyone to sequence their genome in future, yes. So if one is found to be having, if one is having a defective genome, I mean methylation as you said, so for the primary treatment, he will be sequencing his genome, and for the secondary, after it is, many mutations would have been occurred later. So will that be possible for him to again and again sequence his genome? Is that a point there?
Actually, our genome is enlarged, it is going to be constant. It's not going to change. Only if there is going to be a disease like cancer, then only in the tumor it would be changing. So the basic DNA sequencing alone is enough. So in some countries already they started, I think even in Israel there was a project, they were just sequencing everybody in the classroom. Then they wanted to give the sequence to them. Then after 1 year they wanted to see how the behavior is changing, because in all these things there are a lot of ethical issues also. If I imagine, if I have a defective genome, okay, and then imagine everybody tells that this guy has this defect, and then I will not get a girl if I'm not a married person at that time. So those kind of issues are there, and then you can cheat, misuse, and all those things. So ethical issues are also there. But your answer to your question, yes, if you sequence once, that is enough. But if there is a specific disease, that part can be sequenced later. So in large, things are not going to change.
As sir says, you know your stock. What is your genome? Is known to you. You do it once and keep it with you. Then if you want to really look at the intricate issues, epigenetics and things like that, go to him or go to people like them. They would actually use markers and they will give solutions. But it is better we all know our genome. It's now becoming cheaper and cheaper. Okay.
Yeah.
So I'm sure that there will be a day where 10,000 rupees you can get your genome sequence and you can sure. Now that actually helps you to choose the right drug, not to use a wrong drug, not to meddle with genes which are not concerning to you at that point of time. Wrong prescriptions and wrong use of drugs is very rampant here, and that is where this BGM is very very important. You have to contact sir when he is free and have a conversation later. One more question this side.
Yeah, please.
Yeah.
Whether it may be cured?
Muscular dystrophy, I do not know exactly, but okay.
But now are the medicines like stem cells? But now are the medicines like a stem cell therapy may cure or?
But I do not know exactly about muscular dystrophy you are talking about. But there should be some medicines available. Some, I think I differ with ma'am probably. I think if you search, there might be some medicines available. Maybe a temporary cure or whatever it is. I believe that something should be available. But stem cell therapy in eye and something people are like experimenting, and in somewhere it is used. But there should be something available. You can search, and then you yourself can search now. Now Google, everything is there. But I believe something is there. I don't want to say that there is no medicine for that.
Because
I'm sure
this
that
is related to a gene mutation, but how can we cure with stem cells?
no. See, the thing is, see, in one side we are sequencing the genome to understand the disease and pathology and everything. But in another side, we are using chemicals to rectify that. But I am sure that there should be some chemicals which can rectify that. And but because muscular dystrophy is a very common thing actually, so I'm sure that. But a gene cannot be repaired. Repaired gene is repaired gene only. You can't do anything. But you can try to compensate with some chemicals or some proteins which can try to compensate that. But
You cannot change that mutation.
mutation can never be changed. So I think that is very that people have done in Yela Mariu, that is not at all possible. But only thing is, by using some chemicals we can try to compensate. See, the madam was telling of diabetes. Yes, you have some defect somewhere, but if you could understand that, it is fine. But in diabetes we don't know, it is a metabolic. But in this case, it is if it is a gene mutation, it's okay. I think with mutation we can live. See, we all that's what we used to say whenever we do experiment. Who is normal? I consider myself as a normal, but everybody will get one or the other disease. So it is definitely possible to find out some chemicals. I'm sure that it is available. Okay.
Thank you so much.
Do you know madam?
No, sir.
Okay, yeah, madam, you can.
under single gene abnormality. There are 56 single gene abnormality where if only in birth time, if it is identified in the womb of a mother, the gene editing can be done of identifying in the pregnancy. Otherwise, we cannot rectify the disease. Actually, I am working on thalassemia, and my co-scholar is working on this fragile X syndrome and other things, where there is no drug, we cannot be cured. We can identify it and we can reduce the severity.
Yeah, that's it.
Yeah, we cannot, like cancer, if we have a breast cancer, we can cure it.
That is
Maybe. Like,
giving a chemical is a cure, ma'am. If the individually
no,
surviving,
but
it's a
totally we
cure.
can cure now, sir. For example, if you are taking a medicine for fever, it is cured. But for these type of diseases, there is no treatment.
I would like you to be a little bit careful. You're a mathematician. You're going to be meaning a lot to us. But biology is biology. You just cannot go out in a metal and change genetic defects.
Yes, sir.
Now, you can have a conversation with her, and you can have a conversation with sir also. Yeah. Next question.
Sir, I have a question.
Yes.
Actually, I am working on this gene abnormality and I'm doing mathematical modeling. I started my work in 2018 in Tyagaraja College of Engineering, sir. My work was like to be patented. A drop of blood, as you told me, a drop of blood can be given all the DNA isolations, and I'll show all the diseases, the gene abnormality, all the abnormality, that is the device what we are trying to do. But as we are mathematicians, we are lagging there in that first part. The other parts we are completed. So when I keep a drop of blood and I need it immediately to all the RNA sequence, I need the RNA sequence. You told me the kits of COVID has come. Can you explain the mechanism of what is happening there?
But a drop of blood, you want to get the RNA and other things.
Yes, sir.
But I don't know, from a drop of blood, RNA can be whole, RNA can be done. We can discuss, ma'am, sometime. If anything is available in our laboratory, we can definitely use. But from a single drop of blood, of course, there are some methods available for just for diagnosis. Just from a single drop of blood, you can completely get the RNA sequence is not possible. That's what I think.
That was not clear for us in my work. What we were doing is, I was collaborating with Dr. Prabhakaran, professor at Government College of Melo. He will collect some samples of blood, and we are doing as usual the PCR and all. It takes a long process. If you are converting as a device, then as like a pregnancy kit, if you leave a drop of few drops, then we get the result. As like if we leave some few drops of blood, the other part I have completed. Where I lag is only this isolation part. Is there any mechanism?
But there are plenty of kits available. There are plenty of assays, reagents are available. But we'll see specifically what exactly you want. You want to see a single gene, or like what do you want to see? So based on that, we can see, ma'am. So we'll discuss actually. No, we can discuss because from a single group we cannot get everything actually. But because that would be like really going to be very costly thing. Okay. Yeah. Well, that's what I thought. A long time back.
Knowledge generation. Sometimes this kind of weird conversations or meaningless conversations will end up with something very, very good.
Yeah.
Always this transdisciplinary approach is important, but you cannot expect an answer at one point.
Yeah, I know, sir. Yes, I know. Whether our track, what we are going is right or not, I wanted to know.
So we'll discuss, ma'am, sometime.
Yeah.
Discuss with Prabhakar, your guy.
Yeah.
Then meet with him and continue your discussions later. There is another question.
Yeah, you just call me sometime. We'll discuss, ma'am. That's not a problem.
Yeah, thank
Whatever
you so
is
much, sir.
possible, we do without any.
Thank you so much.
What is the current status of Human Genome Project? The US Federation said it was completed in 2003, but in our day-to-day life, we are exposing so many of proteomics works and so many of genes we have identified. So I want to just know that what is the current status of Human Genome Project, whether it is completed or not.
Yeah, Human Genome Project completed around 2006 or so.
US Federation said that it was 2003.
Yeah.
But we are exposing our day-to-day life so many of genes, disease genes, and we discovering new genes every day. So that I just want that whether the Human Genome Project is wholly completed or a partially.
See, Human Genome Project completed around 2006 or so.
Okay.
So the complete genome sequence got openly available and that is done. Then, yeah,
Yes, that is what I just.
For that, subsequently, 1000 Genome Project, so many other things came. Yeah, I think Human Encyclopedia came. But now people are working on working on thousands of genomes. So that is the next phase. And now people are talking about personal genome, not even thousands. Let us sequence everything. So human genome, basically the project got over. Now we are asking question in MSc, what is the output of Human Genome Project, and that got over. But now people are talking about 1000 genome. Now people are talking about million genome project. People are talking about Human Genome Encyclopedia and so many things. So that project got over. Now it got diversified in so many different aspects.
It just expanded its wings.
Expanded. But again, my point, I made it clear, I guess. Genome sequence is known, but functions still we need to understand a lot.
Another question was, is there any space in a Human Genome Project for a diseased genes?
So nothing like a region we have as a disease gene, but sometimes some of the genes which are being useful to us, they are becoming defective, they are becoming not functional, and that is a thing. Or else there is nothing like a disease, but there is something called a druggable genome, something like that. But nothing like a, see, we don't have any region to cause a disease, but there may be some viral dead region, something may be there, but nothing like that. But sometimes our own genes are creating some problem because of wrong expression or so.
Thank you. Thank you.
Thank you.
Have further discussion with PV. Okay, so this is actually the base data is already available, like a Arabidopsis, where we know every gene's function. Human genome has been completely done, but it doesn't mean that you have answers for everything. So take that. That's the best point that
that is what I want to know.
So now that actually reminds me of one of his slides. I was impressed about the kind of genomic work that you are taking up in Kennedy, from the bones, from the teeth, isolating DNA. And in one of your slides, I could see that Punjabis are closely related to Tamils and South Indians, whereas Bangladeshis, they are being shown in different colors. It means a lot to me, and that slide will not be complete with that. There is another slide which Sir was saying that from the African from Africa, how the human migration has taken place. And if you track the human migration, Indians never came into India through Bangladesh route. Okay, you would find that though we were Pakistan, Punjab are closer on the left hand side, the human route, human migration has taken place in that route. Whereas this is something which is standing out, and that is where genetically we are poles apart. Now, Professor Pichappan was actually trying to use genomic data to characterize certain traits of our community, color community here. So such things are there. It's an ocean. Genomics is an ocean. Okay, I'm glad that we are starting it, and I'm sure PV would have learned a lot from Munawar and RJ, and you are with them. So please continue to have this conversation. One last question. We need to go for lunch. Otherwise, we will get scolding from the main office. Okay?
Yeah, please.
Yeah. Who's going to ask?
Yeah, just go ahead, please.
Okay, I'll allow this also. Okay.
I exactly don't know the answer, but I remember it is because of some gene things actually. I don't know exactly. Color blindness.
I got this doubt because I just came across an article which says that a girl got color blindness because she was using a phone for a long time in the dim light, but I don't know whether it is true or not, and and they also told that she was cured from that because initially she was diagnosed with it. So I just had this. No, sir. That was the only article I came across.
Yeah, yeah.
So,
You check in Google, you'll get definitely get it. I don't know exactly. You check that. And also in Aravind, there are some fantastic eye research groups are there, Aravind. But anyway, in Google you will find the answer. All the best.
I'm allowing this question because I have a bias for botany. So there is a botany student, a class opening up. I don't know whether he would
Yeah, please.
be to.
So you know that it will be better if you are, instead of prescribing a particular drug for a particular disease, if based on a person's genome we can give. It is okay if a single person or few individuals are affected. But if, like in case of COVID, if whole of the population is affected, won't it be a tedious and a long process to give drugs for a whole of the population and to segregate them? Won't it be a tedious process?
Yeah, that will be hard, but so we are working on the possibility, everybody is working on the possibility of futuristic possibility. In future, there should be some effective mechanisms. Imagine in the hall, genome sequence is available for everybody, then that is practically possible now.
But like in case of COVID, the whole
Yeah,
of the nation.
at that time we have to forget about everything now. Then, we are drinking all kasayams and everything. So, at that time, this point of time it is possible. Imagine the same thing comes after 10 years. I think we may be well equipped.
The generic prescriptions, okay, they will have its own limitations. But personalized things, sir is moving to the, sir, please continue. So sir is moving to the next level, and he is actually trying to open up your thinking and invite others. But then, sir is neither Brahma nor Vishnu to answer all your questions. He never created or never is going to save you. Okay, maybe he's not a Shiva too. He's not going to destroy you. Okay, but he is a human being, a humble human being like us, trying to learn. He is inquisitive. He calls himself a learner even now. He says that he learns from his students, and he shares the rich with them. So when she was actually talking about patents, I missed a point. Thanks for that. He had already had a patent in his name. So with the group which he was associating in the US, I think on Chinese name also. Okay, so there he has a patent. So here is one person right at your doorstep, Americancollege University. It's not too far away. The thing is, sir, time should permit. Okay, you take some time with them, go through the day, sir. Okay, or any of your friends, and most of the friends here are known to them. Don't waste his time. But with specific questions, go. But before you go to him, go with a net, or at least you would go close to net, so that the conversation will be an intelligent conversation. You should not waste his time, nor you should waste your time. He says that it should be everything is money, but don't go run after money. If it's for the fun of it, for the exploration, you have to do biology. I'm sure that the sir has brought in kind of a different atmosphere to the conference here. We had a long and good discussion. Thank you very much, sir. So it's my pleasure to be seated next to him, a big man, though he's very young in age. I think we need to do some honors for him.
Yes, sir.
So we will go to that.
So now we come to the end of this session. The true warmth of the occasion remains as a hallmark with heart full of appreciation and memories. Today's great moments and are tomorrow's wonderful memories. And of course, mementos or reminders of those great moments. I invite Dr. G.C. Abraham, Vice Principal, to honor our guest, Dr. G. Kumarasan, with a shawl and memento.
I have a person of my age sitting here there. I would request the Sanjay also to join me. Sir, please. Thank you, sir. Thank you very much. Your friend and your is going to be there honoring you. Okay, thank you.
Thank you, sir. And it's time to thank our dignified guest. There is no pleasing exercise of the mind other than gratitude. We thank our distinguished guest, Dr. G. Kumarasan, Head, Department of Genetics, Madurai Kamaraj University, for accepting our invitation and elevating our minds with novel approaches in functional genomics and molecular diagnostics. As science and technology grows by leaps and bounds, and to keep pace with this novel approaches in genetics, drug targets, the challenges should be taken into consideration and be updated. Thank you, sir, for your valuable information and research directions. We remain highly indebted to thank our Vice Principal for moderating the session, and I thank all our faculty friends for partaking in the session, and also thank the students for your very presence and making this session fruitful. Thank you one and all. It's time for lunch. We'll resume back at 2:15 p.m. Thank you.